Hi, today we're going to start to go into the process of creating a DNA profile on somebody. We're going to talk about the old method first, what I would consider the old version, which uses electrophoresis as its main source of separating the DNA. It's important for you to learn the old version before you get to the current method for DNA profiling, because some of the old version is embedded in the new version, and it makes more sense if you've already learned the older part of it. So DNA profiling is a technique used by scientists to distinguish between individuals of the same species, in our case humans, using only samples of their DNA. Okay? The process was discovered by Alec Jeffreys at the University of Leicester in 1985. So it's a little over 30 years old from when they first discovered it, and they've come a significant way since then. So let's get started with the steps. This is the basic process of making a DNA profile. I consider this the old version. Step one, the cells are broken down to release the DNA via extraction. And you guys already did the extraction labs. You did them with both the virtual lab and the banana lab. So you know how to get the DNA out of cells, right? You know that you have to use a lysis solution to break down the cell membranes. You know that you have to add a salt solution to separate out the protein, and then you have to have isopropyl alcohol to get the DNA by itself. And we've gone through that whole process. So step one is to break the cells down to release the DNA. And then if you only have a tiny amount of DNA, you have to amplify it using PCR. And we've talked about the polymerase chain reaction and how to make copies of that DNA that you want as well. So this part you already know how to do. That's step one. Get the DNA out. Make copies if you need to. Then what they do is they cut the DNA into fragments using restriction enzymes. So step two was to take the DNA that they just pulled out of the cells and to use restriction enzymes, okay, like EcoRI or the HA3, the ones we talked about in class, and they put those restriction enzymes into the DNA and it cuts it at the recognition sites, and it makes fragments of all different sizes. Okay, so it makes restriction fragments of all different sizes. And we talked about how we can separate the fragments out according to size a little bit when we talked about restriction enzymes. Step three, and this is the part that we haven't talked about yet, is that they take the fragments and they separate them based on their size using a process called gel electrophoresis. And the electrophoresis is the part that we haven't actually discussed yet. So this is a electrophoresis machine right here. You can see a picture of it here. And this is like the top side of it, or the front side, and this is kind of the bottom side of it down here. The DNA fragments are injected into these little wells. So there's little like rectangles right here. Let's see if I can point to them right there. These little rectangles right here are wells, and the DNA gets injected into them using a pipette. This orange material right here is a gel, that an agarose gel that they have, and they have like little tiny pockets into them called wells, and you inject the DNA into the wells, and then they put a buffer solution over top of the gel. And then what they do is they put an electrical current to go through the gel. So you can see that there's a negative electrode here and a positive electrode here. Well, DNA has a negative charge naturally because of the phosphates in its backbone. Remember we said that DNA is made of phosphates, sugars, and bases for the nucleotides? Well, the phosphate group is negatively charged, so DNA in turn has a negative charge. Well, one negative charge next to another negative charge doesn't work very well together. So the DNA moves down the gel, okay, and through the gel towards the positive side. And the idea behind this is the smaller the fragment, the smaller the piece, the further it's able to move. The bigger the piece, or the longer the piece, the less it's able to go because it just gets stuck in the gel. And so what it does is it separates towards the positive side, basically the smallest one's going the furthest and the biggest one staying closest to the back, or to where it started at. So gel electrophoresis, its job is to separate the DNA according to size biggest to smallest, and they use this apparatus, a gel electrophoresis machine, in order to do that. Okay? And we'll actually take a look at this in class and let you guys try loading some dyes in here and seeing how the process works, how it actually takes the DNA through the gels, and how the electrophoresis machine works. After they run the DNA through the electrophoresis machine, they add a radioactive material called a probe to the DNA. And a probe is a sequence of DNA complementary to the fragment you're interested in. So what they might do is they might add a probe that's maybe TTAAT. Okay, that might be the probe that they add. And it would have a little tag attached to it. So let's see. Let's make it fluorescent. So it would have like this little fluorescent kind of circle bulb attached to this sequence TTAAT. And so what happens is they would add this in, and when the probe is introduced, it bonds to a specific DNA sequence. So if your fragment that was found down here had the sequence AATTA, right? Oops, sorry. Then this probe would bond to it because it has the complementary sequence to it, right? It would bond to it, okay? But when it binds to it, it also attaches this color tag to it, and that basically makes the DNA visible, okay? So the radioactive material called a probe is combined with DNA fragments, and then they put it underneath like a UV light, and then the bands fluoresce. So you can see that this right here, this whole thing was the gel. Here were the wells that it first started in, and it ran through here, right? And so these fragments that are down here are smaller, and these fragments up here are bigger. And you can see that the fragments that we can actually see have the probe attached to it. Now notice if this is row 1, and this is row 2, and this is, sorry, hang on. Let's try that again. 1, 2, 3, 4, 5, 6, 7. I think there's 8 rows. So if these are the rows, okay, and I can kind of space these out a little bit better here. So this is row 1, 2, 3, 4, 5, and then this needs to go a little closer to be row 6, 7, 8, and then 9, 10 here. You can see that some of these match, right? Like you can see that rows 2 and 3 match together. You can see 4 and 5 match together, and you can see that 9 and 10 over here match together, or 8 and 9, depending on what it's at. But you can see that they're in the same locations, right? You can see that there's a band here and a band here and here in the same spots and the same widths. That's how you know that the DNA matches. So these, and then you can see there's like a double band here, there's a light band here, and they're in the same spot. So person number 2 and person number 3's DNA is the same. So like if this was found at our crime scene, number 2, and this was our suspect number 3, you know that it was a match. 4 and 5 are the same, and then these two down here, 8 and 9, are the same as well. So what you're looking for is, you're looking for the same size bands and the same locations to be a match. So step 5 is the pattern of the fragment distribution is then analyzed. So kind of like what we just did. We kind of just did step 5. We took a look to see which bands matched in which locations. The DNA profile is individual. It's highly specific. The chances of 2 people having the same DNA profile is 300,000 million to 1, which is like impossible, right? Except for identical twins. They have exactly the same DNA. Okay, so let me show you some examples of what the DNA profiles look like when you're trying to compare them. So this is an analysis pattern from electrophoresis. So these were run through the machine, and here's the bands. And you can see that this is the suspect's profile. So this is the person in question when we ran their DNA. This was blood from the crime scene, okay, that we found. and then this was the victim's profile. And you can see that the bands match perfectly the blood from the crime scene with the suspect. These two, right, in the same locations. These two both double bands in the same locations. This one's in the same spot. So you want them to be in the same location and the same size. And you can see that these two rows match up perfectly, which means your suspect's DNA was in the blood at the crime scene. So you can for sure put your suspect at the crime scene. Okay, so that's kind of how you read those. And then you can see for the victim, it had a couple of lines that matched, like this one and this one matches, and this one matches, but the rest of them don't match. And in order to be considered a match, everything has to match up. And then for paternity testing for DNA, okay, a DNA profile of a mom and her child, it's possible to identify the fragments that are absent from the mom and therefore have to be the dad's, and then you can see if there's a match. So you can see here, if this is the child right here, okay, the first thing that you want to do is any of them that belong to the mom you want to cross out. So you can see, let me see if I can do a highlighter here. Okay, so the mom, you can see matches here, right, and here on both of those, match the mom. This one doesn't match the mom. This one doesn't match the mom. This one does. This one belongs to the mom, right? And this one doesn't. So now those three belong to the mom. That child got that from the mom. So you know the ones that are left have to match the dad in order for it to be considered the dad. So this one is in the same location as this one. This one is in the same location as this one, and this one is in the same location. So this guy is the father of this child because the DNA that was left over that we subtracted out for the mom matched the dad perfectly. And you get half your DNA from your mom and half from your dad. So when you're looking at a electrophoresis printout of a DNA profile for a child, that's how you do it. You subtract out the mom's bands first, and whatever's left over has to match the dad in question. Okay, so we'll do some practice at reading the electrophoresis printouts and getting comfortable with making matches with the electrophoresis, and then we'll go to the current DNA profiling process. It's kind of tweaked a little more specifically.